Hello, welcome to today's webinar, Forging Strong Links for Patient Safety, the Power of Implementing the Chain of Identity Identifier. This is an ICCBBA webinar and involves the ISBT 128 Standardized Chain of Identity Identifier. In full transparency, I am recording this introduction after the live event because I encountered some serious audio malfunctions during the live event. Fortunately, I was not one of the primary speakers. So just have a few things to cover beforehand, and then the rest of the content will be as was presented. So what is the ISBT 128 Chain of Identifier? Robin Wilkinson, the Deputy Technical Director at ICCBBA, will be presenting that information. Then we'll follow with someone from the manufacturing sector. Aunt Beth Gardner, who is the Senior Director of Patient Supply at Beam Therapeutics, will talk about the benefits of implementing the ISBT 128 standard label. That's the hybrid label, and some challenges from the manufacturer sponsor perspective. Catherine McIntyre and Neri Diaz from Stanford Medicine Cellular Therapy Facility will talk about benefits of implementing the ISBT 128 hybrid label and the chain of identity identifier and some challenges associated with that. And then there's a great panel discussion where we can answer your questions, that sort of thing. And this will be moderated by Alex Garlitz, at our ICCBBA Information Standards Specialist. Please submit your questions at any time during the webinar. So how to submit questions? Just look up at the top right-hand side of this screen. Make sure you're on the live stage and you should be able to see the Q&A feed. If you don't, you might, and if you've not been to one of our webinars before, you might have to acknowledge a message that comes to you, but it's pretty straightforward. All right, what is ISBT 128? ISBT 128 is the international standard for the transfer of information associated with medical products of human origin. Anything that comes out of a human being to be used medically in another human being can be labeled with ISBT 128 standard. This ability to bidirectionally trace that is provided by the standard, to bidirectionally trace to and from the donor recipient, is essential for global biovigilance of medical products of human origin. The standard provides standardized terminology and globally unique donation event identification. What about ICCBBA? Well, ICCBBA is the nonprofit that was established in 1994 to develop and maintain the ISBT 128 standards for terminology, unique identification, labeling, and electronic data exchange for MPHO. All our standards and implementation guides are developed with the aid of volunteers via our technical advisory groups, and you can look at our website to learn more about that. And a reminder that ICCBBA is indeed an international nonprofit, non-state actor supported by user fees. Next up, we'll have Robin Wilkinson, our Deputy Technical Director, speak about what is the ISBT 128 Chain of Identity Identifier. Thank you for your time.
Thank you, Karen. Hopefully the technical difficulties will not continue. We're going to go ahead and proceed. We are going to start with some definitions. These were developed with the standards coordinating body and with ICCBBA's Cellular Therapy Coding and Labeling Technical Advisory Group. These definitions are also consistent with AABB and FACT JC standards and are provided in ISBT 128 Standard Chain of Identity Identifier ST028. It's the CoI Identifier solution that we're going to focus on today. The ISBT 128 Chain of Identity Identifier is a unique, meaning globally unique, end-to-end code used to identify the cell or gene therapy that enables a bidirectional link between the donors and the intended recipients. I want to take a moment to discuss the need for the ISBT 128 Chain of Identity Identifier. The ISBT 128 donation identification number provides a globally unique identifier for a single donation event, as well as the products derived from that event. But circumstances exist in the collection and processing of some cellular therapy products where more than one donation may be needed to deliver a given therapy. In these cases, the individual DINs of multiple collections need to be linked, as well as the downstream products or doses made from those multiple collections. Therefore, a new identifier was needed, which is the Chain of Identity Identifier. Now, this was not a new concept, but people were using many different formats, lengths, and the possibility of duplication of those Chain of Identity identifiers existed. So ISBT 128 standardized the structure and format of the CoI identifier, and that supports patient safety, efficiency, and interoperability between software systems. The CoI identifier does not replace the DIN on collected products, but will instead be an additional identifier that is the same on all collections and subsequent products or doses associated with a given therapy. You'll notice that the DIN and the CoI identifier do look a little similar, but they have slightly different formats, which we'll talk about in a little bit. First, let's talk about the structure of the CoI identifier, which is ISBT 128 data structure 040. Every ISBT 128 data structure begins with a 2-character data identifier to indicate it is in fact an ISBT 128 data element. The data identifier for the chain of identity identifier is an ampersand and /. Following this, we have the fixed text CH to help identify that it is the chain of identity identifier. After this, we have a facility identification number, or FIN, which is an alphanumeric 5-character identifier that's issued by ICCBBA to registered facilities. And ICCBBA maintains a database of these facility identifiers to ensure global uniqueness. And after the FIN, we have a 2-digit nominal year code indicating the year of assignment. And finally, we have a 6-character alphanumeric sequence. This sequence is maintained by the assigning facility and must be unique for the FIN within the given year. Through the combination of these elements, the CoI identifier for the given therapy is now globally unique in a standardized format. When printed on a label, the CoI identifier is accompanied by a checksum character to help ensure accuracy when manually entering it into software, such as when you're typing it into software from a keyboard. The checksum character is not part of the data content of the CoI identifier, and so is not included in the data structure or barcode. This is because it's really not intended for use when the CoI identifier is scanned electronically. The checksum is actually calculated via an algorithm on the 15 characters of the CoI identifier. And note that the 2-character data identifier, ampersand and / are not included in the calculation. And I mentioned that a little bit before that the DIN and the CoI identifier are structured similarly. This is because this format, turns out it works really well to establish globally unique identifiers. Significant differences do exist, however. For example, while both the ISBT 128 DIN and the CoI identifier contain a FIN, a nominal year, and are accompanied by a check character, the DIN is 13 characters, while the CoI identifier is 15 characters and begins with that CH. The sequence in the DIN is numeric, while the CoI identifier sequence is alphanumeric. And finally, DINs are accompanied by flag characters, which are used to identify specific items associated with a collection, whereas the CoI identifier is not accompanied by flags, excuse me, since they are not needed. And then once again, the DIN identifies a specific donation event, whereas the CoI identifier links multiple donations to a patient-specific given therapy. Here is an example of a hybrid label for a product collected for further manufacturing. Detailed requirements for this label are actually documented in ST018. That's the ISBT 128 standard labeling of collection products for cellular therapy manufacturing. The left half of the label has the ISBT 128 data elements of the DIN, the product description code, and the division identifier. And then the right side of the label will contain the CoI identifier. It's noted there in the green arrow, as well as other data that's based on some if-then conditional rules, which are outlined in ST018. I should note too that the ISBT 128 chain of identifier can also be used on non-ISBT 128 labels if so desired. Additionally, ISBT 128 is not just a labeling standard. It's intended to facilitate interoperability between electronic healthcare systems. In ISBT 128 Data Dictionary of Standard Data Elements, which is ST027, and ISBT 128 Standard for XML Messaging, which is ST020, we discuss data elements and unique resource identifiers, or URIs. I won't go too deeply into this, but essentially URIs are like the data identifiers in the barcode, but for electronic messaging. ICCBA has assigned each ISBT 128 data element a URI for identification in the form of a URL, which are innately unique. Here we have an example XML on the right, where you can see how the CoI identifier URI is used. Lastly, here's a diagram that depicts the use of the CoI identifier to link elements in a multiple collection to a multiple therapeutic dose scenario. Each collection, which could be from a variety of MPHO sources, will be assigned a unique DIN. The CoI identifier should be assigned prior to that first collection, and the entities involved in the given therapy should have an agreement in place on whose FIN should be used within that CoI identifier. That same CoI identifier is then assigned to each subsequently collected product, in addition to a DIN that is unique to that collection. Then the same CoI identifier is assigned to each manufactured product and therapeutic dose throughout the life cycle of the biological material. Finally, I have a summary slide to show you the information that I presented today. The ISBT 128 CoI identifier is a globally unique end-to-end code in a standardized structure and format that identifies the cell or gene therapy and enables a bidirectional linkage between the donors and the intended recipients. I hope this information was helpful, and thank you for your time. I'm now going to hand it off to our next presenter, which is who's Neri. Thank you, Neri.
All right, thank you so much, Robin. Good morning, everyone. Before I get started, I wanted to mention that if you have any questions, please use your the chat window under the Q&A section, and those will be addressed at the end of the talks for today. So I'm here to talk about our experience at Stanford Healthcare and the benefits and some of the challenges that we've had during implementation. So we're located in the Bay Area, so there was no way I could pass up the opportunity to share a less common picture of one of our icons, which is the Golden Gate Bridge, and I think is a great example of how chains are so important to our safety. So some of the benefits of using the ST018 label during your care of patient products is that it includes a lot of standardization. The information is clearly readable and understood, and all of the established rules and processes are easier to follow, train, and enforce than coming up with your own system, essentially. There's a great element of safety. It prevents the duplication of product and patient identifiers. There also is the offering of consistency. There's convenience as well. So we're going to be using current labeling hardware, software, and consumables. You're not going to be having to purchase a lot of additional items. And you're essentially repurposing or using a format that already exists. And this is really important because all of us in the cell therapy community understand that there is an increased demand for commercial products as well as all kinds of different trials. And another excellent aspect of using the ST018 label format and chain of identity is that it is recognized internationally and accepted internationally. So all of those benefits do come with some challenges. One of them is that you do have to invest in education. And that's where all of you have taken that first step by being here today. The standards are not always easy to implement, especially if it's the first time you've read them. Receiving guidance and collaborating with other people or centers who have used these standards is a great benefit and is really critical to having success. And another challenge is that the users of the standards will very rarely have both a cell therapy and an IT background. So getting those two elements to work together can be challenging. Another challenge we find is software updates. So as we know, every time there's a new product being added or a new product code, that may require an update. Sometimes you'll have to find a way to do a manual entry until the new updates are available. And validating your system every time there's an update can be really daunting and time consuming. We've also found in our own experience that different types of software can provide different results based on how updated they are. So you might have very similar products, potentially even with the same vendor, that behave differently if they're standalone versus web-based. And another difficulty that we're all experiencing is an increased need for cybersecurity. And that can really slow down installation and subsequent updates. And that is definitely something to be kept in mind. Another difficulty or challenge is jargon, the words we use to achieve consensus. We all have to be speaking in that same language. Even the concept of CoI, if you do not say chain of identity, could lead to a misunderstanding. CoI may have other interpretations. And sometimes you're really close, every single element is perfect, and you'll find one thing that makes you non-compliant. So that can definitely be frustrating. We also find that it is really critical to have that conversation with all the sponsors and manufacturers. Some of these companies may only be familiar with pharmaceutical regulations, for example, because that's been their primary connection to the medical community. Sometimes they could also be unaware of the fact that there are cell therapy regulatory bodies or requirements, or even if they do know that they exist, they may only have a very basic understanding. And also many times, especially with trials, they may already have software or systems in place that aren't taking this into account. So that provides a barrier that we have to try to get over. And lastly, one of my biggest frustrations, which is when we have all of this labeling and everything so beautiful and set up, then the FDA approves a product. And when you find and you get the label, you realize it has nothing to do with ISBT 128. But at that point, it's too late. We can't do anything about that. So, all right. So how do we encourage other centers to take on these challenges and actually get over these barriers and actually start to use this new standard? So we have a couple of strategies that we've been using to encourage the adoption of the CoI chain of identity identifier. The most important step is to talk before you onboard. So we've developed a scope of services document that we disseminate to our PIs and sponsors that draws attention to the use of ISBT 128 standards. We educate the sponsors on the requirements, and we also make sure that we educate them on the fact that per the California Department of Health, ISBT 128 is a requirement. Setting the expectations with the sponsors in advance is critical to allowing them to be open to accepting this standard. And we have an agreement, unless there's an agreement in advance with the PI, that there can be an exception. So one of the things we've taken to doing is checking our ICCBA tool to make sure that our sponsors, our facilities, are sponsored with ICCBA. And we also have to do ongoing reminders and ongoing education as things continue forward. But getting that communication in advance is definitely the huge win in terms of getting this to move forward smoothly. Okay, and with that, I will conclude my section of the presentation. I will now pass the baton over to Beth. Thanks, everyone.
Miri, thanks so much. Hi, everybody. My name is Beth Gardner, and I am Senior Director of Patient Supply at Beam Therapeutics. And today I'm going to talk about how sponsors or manufacturers can be a catalyst for this change. right? So we've shaped and we've created this new standard that allows for consistency for data integrity, for patient safety, as we cross the ecosystems especially for autologous cell therapy, right? We're collecting cells from one location. We need to move those cells to another location for manufacturing. And change is hard, right? Change takes time to implement, and we're in a transition period. So I'm sparking everybody to think about how we can be that catalyst to accelerate this change. So as a sponsor biotech manufacturer, when I was thinking about going into each of our clinical sites to ask them and set up to be part of the clinical trial, I realized I wasn't alone, right? We have an amazing landscape of cell therapies, and gene edited therapies that are coming to potentially be tested on humans, right? And so here's a landscape of cell therapies by Andrew Panu, who's now part of Sleuth. This image here is from earlier this year, and the link is on the right-hand side there. But to orient you, the outer ring is preclinical. And as you go inside the circle, you start to see products that are phase 1, phase 2 to phase 2, phase 3 to marketed products. And that there's a legend, and there's a subsequent slides here that will show you. So each legend is a little bit different between autologous, allogeneic, and sort of the approach for the therapy. So by no means was my single product, was I alone, right? I was having to walk into respective clinical sites and ask them to do procedures that weren't just for my company, for the patients participating in my trial. I had to make sure, and I really had a guiding principle to say, how could we make the processes that my company was asking for to be as consistent as possible with industry. So here is, as I mentioned, cell therapy. Here's an image for gene therapy products. And then here is an image of gene edited products. So I mentioned, so I tend to fall to this concept of guiding principles. And if I'm a pharma biotech manufacturing company, if you're clinical supply chain, or what my organization is called is patient supply, right? The clinical supply chain often becomes the commercial supply chain. So instead of calling things clinical or commercial, I called it what the purpose was for the patient. And so establishing these guiding principles early was really helpful to help cross-functional stakeholders understand the purpose of implementing ISBT 128, Standard 18, and Standard 28. And so for me, I wanted to create the lowest burden possible for the patient, right? We were going to create access to medical centers to provide clinical trial for patients with unmet medical needs. I wanted to make sure that we hit the target, the lowest number of cycles for apheresis as possible, while establishing the highest quality cells, right? I don't want a patient to be on an apheresis machine any longer than what's absolutely needed for the manufacturing process. It's not an easy procedure. And so with this, then, how did we utilize these new industry tools, right, to ensure data integrity and also then connecting patient safety? Lowest burden to the site, right? So my goal was not to have to go in to do specialized audits, right, if sites were FACT or AABB accredited, right? In FACT accreditation, AABB, you'll see that there are references to the ISBT standards. By establishing those things, I knew that my sites were well established and could be compliant. I didn't want to provide any special sponsor IT technology like labeling printers, right? Or label stock, right? The clinical sites are not warehouses to be able to store these things and to have specialized systems for every single company. Can you imagine, based on those landscapes, if there was a printer for every single company? It's just not scalable. My goal was then to also create a process where the site generates the donation identification number, right? Normal processes. And my company, Beam, would generate the chain of identity identifier. And so you'll see as I go through this, like, how did we implement this, like to give some step-by-step guides, there's different ways to document this roles and responsibilities. An example that I'll share is through training, right? People are assigned training for apheresis or through the cell therapy lab. And by making that statement, my company will generate the chain of identity number. You're documenting that discussion and that agreement and that decision. The last piece here is I wanted to anchor. I wanted to anchor to an industry standard so that when it came time for our BLA to create a marketed product, I really had a leg to stand on to say, here's what I've established consistently across all of my sites to ensure patient safety and data integrity. All right, so the nuts and bolts, like how did we implement this? One step at a time. Yeah, Beth, okay, that's obvious, right? Like give us a little bit more detail. Clearly, right? These are the slides that are coming up. But if you break this down into 5 steps, it's a little bit more manageable, right? It's palatable for someone to approach this. And so let's start with step number 1, learn, right? So I always believe in an educational mindset, and I'm a sucker, right, for reading lots of different things. But if you're going to talk about being standard 18 or standard 28 compliant, you should read the standard. It sounds obvious, right? Coming to a session like this is really helpful, right, for an overview, but it's really valuable to actually read the standard to make sure it's applicable to your product, to your company, like how to implement it. And so for easy access, I've included 2 QR codes here, right, for standard 18 and standard 28. And little pull-out photographs, images, right, of what the 4x4 label looks like, right? I was part of the committee that established these 2 documents, and the feedback from the clinical sites were, don't touch the left-hand side of the apheresis label. It's like muscle memory. We know upper left is where the DIN goes. We know that the middle left section is the product code. If you want to add additional information for the next ecosystem, like the manufacturing site or the cryopreservation location, whatever that might be, use the right-hand side of the label, right? So the team created, here's an example of all of the possible identifiers that could be there to include so that as it left your site ecosystem and goes to the next one, they're able to pick up and there's no loss of information. We maintain that chain of identity, right? We maintain that chain of custody. Standard 28, right? It's, again, it's really useful document to read. Start with the table of contents, right? Screen through those sections, and then you'll find like, okay, it's actually the number similar to a DIN is broken out into sections, right? CH, chain. It's not CI for chain of identity because an I can look like an L, can look like a 1, right? So the team that created this standard established CH for chain. The issuing organization, so in this case, Beam, this is Beam's facility identification number, W4596. If you heard me speak, I joke that this is tattooed somewhere on my body. It's not, right? But it's unique to Beam, which allows any number, the last 8 digits that are issued afterwards, to be globally unique for Beam. I know that if this number comes back, I can search to understand which product it's for, what year it was issued. So this is where the control for a sponsor biotech manufacturing company comes in, right? Register for your facility identification number. Similar to a DIN, right? Using the same nomenclature, lexicon that we're all used to, right? Year of assignment, in this case, 24, right? We're in 2024. And then the last 6 alphanumeric digits can be determined by your company, right? Maybe you have product 1, maybe you have product 2, maybe you have product 3. So you want to have those, the first digits kind of assigned based on that. That numbering can only be determined by your company internally. And because of the alphanumeric, you're allowed, it creates a lot of really great permutations, right, to make sure that it can be product specific and then unique to the patient that this is issued for and also the therapy, right? Those are the two things for Standard 18 that we need to make sure the chain of identity identifier and through Standard 28 is issued to a patient and their therapy. All right, so now you're subject matter experts, right? Well, now you need to share that information and you need to align with some stakeholders. So if you're a sponsor biotech company, there's groups like regulatory, clinical operations, your manufacturing team, right? Like you're shipping these cells, right, to another group that has to then forward process it. And so here's some examples, right, for the biggest lessons learned that I can share with you so that you don't repeat the same mistakes or delays that I may have experienced in my career, right? The IND. So this is a new drug application, right, that's submitted, and it includes a drug product label, right? So not the apheresis label. After it's been forward processed, you create a drug product label. But the FDA does not include any requirements currently, right, for autologous cell and gene therapy products to include a chain of identity ID number on that drug product label. And I think that's caused some problems for certain sponsor biotechs. You heard it from Neri, right? They're coming in, they've already submitted the IND, they're doing their training, they're excited to get their cell therapy products right to patients. And when they talk to the apheresis or cell therapy labs and start to talk about identifying numbers, there's a DIN number, there's a product code, there's an option for a CoI ID number, but the sponsor biotech company doesn't have that linkage, right? Because they haven't thought about that because they're only thinking about the end, that drug product label. So to be a catalyst, you need to think about the drug product label and the CoI ID number to be issued well before the DP is manufactured, and to include that sort of within your proposal, right, for IND. The other thing here is if you did do it during clinical development, you can't include it within your BLA, right? Or it becomes much more complicated to do so. So I always say adopt this standard early and utilize it often, right? Include the CoI concept in clinical, and this will enable you to have this option, right, once you go and become a marketed product. All right, what are some other questions to think about, right? So we've all mapped this out, right? This is, a cell journey, a patient journey, right, as an example here. And we often talk about this upper, section here as a clinical site as like one thing. But if you're part of a clinical site, that's not the case, right? You're not one person doing lots of things across all of these parts of the organization, right? There's a group of people who may be working on the screening activities for a patient. You have another team like an apheresis collection center, right, that's working on cell collection. You have a warehouse receipt team that's working on drug product delivery. You're working on a transplant team that's working on, giving maybe a chemotherapy medication for transplant. You have another team. So again, how do we connect the dots to make sure chain of identity identifier goes through not only each of the departments within the clinical site ecosystem, but as we move through these chevrons and go to the manufacturing location, that we're taking that into consideration as well. So some things to think about. What language should be included within the informed consent document, right? HIPAA and GDPR, it's about giving power and permission from the patient, right? If the patient, the human, gives permission to utilize their data, then you're able to share that information, right? So how do you inform the patient how their identifiers are being utilized, and therefore they have awareness and they're informed and they can give permission to do so, right? So consider that language within your ICF document to allow chain of identity, perhaps first name, last name, date of birth, right? So that sites aren't having to black out information, right? That it's shared within a secured way or location within that ICF document. Technology strategy, right? So this is thinking about for manual COID, absolutely. This process for standard 18 and standard 28 does not require an electronic system, right? This can be done through controlled ways, through manual processes. But if you're talking about scalability, if you're talking about volume, how long do you want to maintain, or how long should you maintain that manual process, right? Versus switching over to maybe an electronic system. And then if you're utilizing an electronic system, is that vendor an approved vendor for ISBT 128, right? Part of ICCBA, right? And you can go to the website to see the vendors that are registered, right? And part of ISBT 128. If you find a vendor who's not, have that conversation. Maybe they are following the standards, or they're utilizing the format of the label, and they need to register themselves. So I highly encourage all technology providers out there, if you're utilizing the standard and you're utilizing any parts of the labeling format, that you must be registered with ISBT. Because I'm looking for it, right, from a sponsor biotech. As part of a supplier qualification, it's one less step for me to have to evaluate if you're already an approved supplier. So you're making it a bunch simpler for me and being compliant. All right, operational alignment, right? When is the chain of identity number assigned? Is it assigned at time of cell collection? That's the requirement, right? Prior to cell collection? Absolutely, that's a possibility. Is it registered? Are you creating that number much further upstream at part of screening, right? So a patient enters into the trial and they're issued a chain of identity number. Absolutely, right? So you just have to talk this through with your counterparts to ensure that that those pieces and alignment is in place. And then the last piece here is, I know Neri has spoken to it, Robin spoke to it, but there has to be ownership of who is assigning the chain of identity identifying number, because it links to that facility identification number that's part of the code. Who's issuing that COI ID, right? Document it, have alignment and agreement and clarity to the sites and to your cross-functional stakeholders for who does that. So in my case, Beam is the one to create the chain of identity and issue the chain of identity identifier. So the issuing organization, Beam, we utilize our FIN number. And the DIN, the donation identification number for each collection, is assigned by the site. All right, how do you register for a FIN? Out of all of the steps I've just described, this is the easiest, because ICCBA ISBT has made it the simplest process possible. So here's a QR code if you need to register your FIN number. Here's an example, right, of the landing page that you'll find, along with like some really nice, easy steps to follow, right, to become a facility, to have a registered facility identification number. I may apologize to ISBT ICCBA because these images are from 2023, but nevertheless, conceptually, right, on the left-hand side here, give information about your company, right? What is your legal entity name? What's the name and address? Who are the points of contact? The points of contact that are listed here will be the ones to receive notification for payment or for annual updates, right? Because this expires on an annual basis. Very simple. Out of the super long document, there's just one section here, right? Chain of identity identifier. Click the button. Go to the next page, right? And then payments, right? So for this number on an annual basis, it's a very low cost, right? And so again, ICCBA, ISBT, right? They've described the organization that they are, right? The funding that's received based on these applications, right, goes to kind of the payment of the organization and the very small staff that's maintained in order to create these globally unique numbers. That's it, right? So there you go. I think the longest step that took me is just to verify that I had the correct legal entity name, right, for my company. And that was a quick email to the legal department to assign that. So it doesn't take very long, and I think it's a simple process. All right, the last 2 steps here, right, out of the 5 steps. So step 4 is document, right? So document your chain of identity identifier, the labeling, maybe your cell journey, right? Map out those ecosystems. Personally, I'm a multisensory learner. I can be spoken to, I can visualize different slides, and read different documentation, right? But until I actually talk through or do an activity, it doesn't sink in as much. I suspect a lot of us are that same concept for a learner. So I always encourage multisensory learning and to have those whiteboarding sessions, have those discussions, right, to talk through those steps. The last is, you might, as a sponsor biotech company, if this is a brand new process, consider doing an FMEA risk assessment. This will outline all of the possible things that could go right, go wrong, and everything in between to make sure that you're mitigating and determining that risk for mitigation for each of those steps. And the last thing I'll speak to here is around patient privacy, right? So before we get into that, we've talked about how we've done education, right? The standards are in place. We've done education. And so now what we're doing is we're in this implementation phase, transition phase. But let's be real, right? Either not every sponsor biotech company is going to be ready, or not every apheresis cell therapy lab has updated technology, right, with the new labeling template available to them. So as a sponsor biotech, one of my other guiding principles that I mentioned is how do we remain dynamic and flexible, right? Because we could have top tier sites that either are in the process of implementing the standards and weren't quite ready yet, right, by the time of clinical trial start, or there was other steps that had to be considered as part of their processes. So for me, the important piece was linking the donation identification number to that chain of identity identifier, right? I needed a single location where both of those ID numbers were in one place to prove that the collection bag is linking to this patient and this therapy. So there may be cases, not ideal, right, where a site is utilizing a standard ISBT 4x4 apheresis label, and we will provide a template for a supplemental tie tag label. Again, I'm not providing IT technology, I'm not providing labels. So this means that the site will have to utilize locally, right, a tie tag, and let's say an Avery label. But by having that label then affixed to the bag, I'm able to meet my guiding principle requirement to connect chain of identity number to DIN number all on one label. The last piece that I didn't speak to, and I apologize, I must have taken this slide out, is around patient privacy. So all of us, if you're an apheresis nurse, you're actually speaking to, you are the nurse for that patient, right? You know who that human is. Their name, date of birth may be on a label, right? That's listed here on the left-hand side, right? Or on the actual apheresis bag. And as it moves through shipping, right, through a courier to the next ecosystem, I was very conscientious to ensure patient privacy. And so we generated and talked to a number of stakeholders to ensure that we were honoring the patients, right? They're donating a part of their body, right, to create a medicine. And so how do I ensure their privacy, right, from a patient perspective? And so we created policies and guidelines, right, for our internal employees, and also then worked with our contract manufacturing organizations, if we utilize them, or our contract research organizations that may have access to that data to be able to ensure patient privacy. So I encourage you for that final step is to, again, pull everything together to ensure that once you have this chain of identity number, you're allowing those data elements to move from one ecosystem to the next, that you're ensuring patient privacy. So again, I think it's very difficult, right, to be a catalyst, right? But we've been gifted to be able to have these standards forged for us. And I just wanted, before I end, right, everyone can read the slide there, but if there are questions, go to the ISBT128.org website and click on the button for help. The support desk is amazing, and as you have questions that come up for implementation, if you want someone to scan a label to see if it's what is expected and meets the whatever those examples are, reach out, right? ISBT has just been incredibly a great resource for me, and I just wanted to make sure that you also knew about that resource available to you. If we're not connected, I hope we are soon. Here's my information, and thank you so much. So I'll turn it back to Karen.
Thank you from Beth, and I thank Beth and Mary, and I welcome Catherine McIntyre, who's also from Stanford. Robin's coming back. I'm going to introduce Alex Gartlitz, who's one of his duties is to help wonderful support that ICCBBA provides.
Karen, I hate to stop you, but you're a little garbled again. So
Okay.
Basically what Karen was stating was that the NMDP that's highlighted here in the middle of the slide has developed a validation template to assist you in implementing these standards and your labels. And also I want to reiterate that we are ready to assist and help you with anything that you need that we can provide answers to. As Beth indicated, please contact our help desk. It's support@isbt128.org. And now Alex has some questions ready for us to ask and answer.
Yeah, thank you, Robin. So we did have a few questions that came in during the webinar here. And just a reminder to everybody still in attendance, if you did have any further questions, you can submit into our Q&A section on the right-hand side of your screen there. And if we're not able to get to your questions today, we'll be able to address them at a later date. But the first of our questions that we have for our panel is, who is responsible for allocation of the CoI ID?
Beth, would you like to take that?
I can start, right? So I think the standard itself allows flexibility, and we did that intentionally. So the CoI identifier can be issued by any issuing organization. So whomever might have a FIN number, that could be the sponsor. So in my case, right, as I described, and I think this is more typical, right? You're seeing the sponsor manufacturing company be the issuing organization for a chain of identity ID number. But that's not to say, should you choose to, that you would like to have maybe the apheresis or hospital system utilize their FIN to be the issuing organization. We've spoken to organizations like, that have investigator studies. And perhaps in those cases as well, if you're a research institution, right, that's part of a hospital system, it makes sense then to have the CoI ID assigned by the hospital using their FIN. Neri, Catherine, would you agree with that approach?
Absolutely. It's really important that this is worked out in advance so that everyone understands who's going to be issuing that number. I give you a real life example is that we sent out a product to a sponsor, and then we received it back from that sponsor, and they've used our FIN number and then changed the end number, and that happened to be the same as a patient that we'd already assigned that number to. So that's and the whole point of this is to make it unique and not get mix-ups. So so it's really, really important that that is worked out and that everyone understands and follows the same methodology. We as a clinical site, we would prefer that the sponsor would assign the CoI, but we are open to doing that differently as long as everyone agrees in advance and it's not a surprise.
Yeah, Catherine, and you sparked something too by, I also think if you're part of a clinical trial, there might be a feasibility questionnaire or discussion that occurs in advance. And that's a really great way as well as to get a reading from the sites, right? Are they okay with having the sponsor biotech be the issuing organization for CoI ID, or would they prefer to be the issuing organization? Yeah.
Yeah, absolutely. Just because we prefer it one way doesn't mean that another cell therapy facility likes the same way. Yeah.
Fair. Yeah.
Awesome. Thank you guys so much for that response there. We're going to go ahead and move on to our second question. And that is, must issuing organizations register with ICCBA? And I'll actually go ahead and address that one since that's already kind of been touched upon from our speakers here. Since a facility identification number is a component of the COI, it would require a issuing organization to register with ICCBA. Once you register with ICCBA, ICCBA allocates you a globally unique FIN that identifies your facility. All right. Anything to add from anybody else on the panel on that? Oh, perfect. All right. Then let's go ahead and move on.
I have something to add. Yes, absolutely.
Please.
You might make a number up that looks just like an ISBT 128 FIN number, and it with the very, very best of intentions, but somebody else might already have that number. And so that's why you really need to register. And we've also found that as well.
Yeah, and it could be as, first of all, that's really scary, right? Because if I find somebody else using a W4596 code, like we're going to have some conversations about that, because you're putting my patients at risk, right, who are part of my study by copying my number, which is against the whole process here. But let's say you have generated a number. I think that's where the help desk for ISBT can come in. Like perhaps if that number hasn't already been issued and you also are generating, like there's just, I think, some conversations that could occur around that. And I'm making things up on behalf of ISBT, but it's really important. It's a must-have, a non-negotiable, right, that the issuing organization must be registered with ICCBA.
Yeah, and we routinely check that now. We were burnt by that, so now we always go and check using the tool that Neri mentioned.
Awesome feedback. Thank you very much for that. We're going to move on to our third question here. And for our panel, if a facility's software does not allow inputting an ISBT 128 compliant COI ID, how can the COI be applied to the product? I'll leave that open for our panel to discuss.
So we've certainly come across that issue as well. And I think the key word here is flexibility. So it's important to open dialogue with the software manufacturer to let them know that this issue has come up and that they may need their help. But as long as you have that communication with your sponsor, there are various methods to address the problem. You could use a tie tag, as Beth demonstrated in her presentation. It's possible that you could, even though it's frowned upon, handwrite the COI number onto the label. But the key here is to have that open communication so that everyone is in the loop about how it will be addressed. And hopefully those issues will resolve themselves over time, but they are a part of this growing process.
Yeah, and Neri, I know we talked yesterday a little bit for the pre-meeting, and I give the example of my handwriting is terrible, like really bad, like chicken scratches, right? And so if I were to be the person to write the COI identifying number, right, with 15 characters, right, that are there, like I don't know if that's readable. And so the idea is best, but it would still be compliant. Right, so the best way to do it is to be able to utilize the new standard 18 label, right, 4x4, to have that there. If it doesn't, then I think that deserves a conversation, right? How is there a white space that can be utilized? Is there a hang tag that can be utilized? But again, the goal is to have a single label that connects the DIN number, the COI ID, right, to show that this collection bag belongs to this patient and this therapy.
Awesome. All right. Thank you very much for that. We are coming close to the end of our time here, so we're going to go ahead and address one final question while we are live. And I just want to remind everybody else, if we didn't get to your question today, we will be responding to them at a later date. So keep your eyes out and open for that. So our last question that we're going to be addressing here was one that's directly to you, Beth. But if our panel members would like to jump in, more than welcome to, of course. And the question is who is actually printing the label? Is it the sponsor or the site? And if it is the site, how is the COI transmitted to the site?
Yeah, great questions. So I mentioned that for my sponsor company, we're not providing any IT technology, so no printers and no label stock to the sites. And I think that's been received like a breath of fresh air.
Yes.
Right. Yeah, they don't have to have a warehouse to store all of these specialty printers.
Yes.
So as a result of that, the site is printing the label. We've identified sites that are already standard 18 and standard 18 compliance that can print the 4x4 label. And for those that are not, that we may have a hang tag label, we provide the template that's pre-populated and it's been verified through internal procedures, right, to verify COI and patient ID numbers and all the details that are necessary are validated and correct. And that's sent via PDF to the site to be printed. So that's just one example, right? Again, it's how do you describe and ensure that control? And for me, again, because I love IT things, but I'm also, you know, I work from home and have cats that may walk across the keyboard occasionally. So Word documents, sending it to somebody, there was a chance to be edited. So we wanted to ensure that it was in a PDF format that would be much more difficult to edit.
I think it would be really great if when you're transmitting that COI to include the barcode format, because we know a lot of centers are already scanning barcodes, and that's a great way to apply the chain of identity without worrying about how you're typing it in. And I'm going to also take this opportunity to point out that Karen was being our model and showing a sticker, and I will let Robin explain what that's about.
So we would like to continue this conversation. ICCBA will have a booth at AABB coming up in October. And we have these handy dandy stickers to give out if you've implemented, or if you just want to continue the conversation with other people, sport our sticker and continue the conversation.
So we want to thank everybody for joining our webinar today. Thank our guest speakers for joining us. And again, keep an eye out for our responses to any of the questions that we didn't get to address today. We will be posting those on our website very shortly. And we hope everybody has a wonderful rest of your day. Take care. Bye-bye.